I know him from our my participation in the Cactus and Succulent Society, which Tristan's also a member. But Tristan's got an interesting story from a big picture book of colorful South American birds that he received when he was in kindergarten. His interest grew and his curiosity drove him to visit more libraries, to seek out more bird books, and inspired his passion. And you know, from past presentations, I love the story of the passion in that. But in the 7th grade, Tristan wrote to the head of the ornithology department at LSU requesting inclusion in his next South American field research trip.
Wow.
And although the letter didn't result in an invitation, Tristan continued on with LSU, attended LSU, and also Kansas, University of Kansas, and then started to join different expeditions to South America, China, New Guinea, and I think the Philippines. In early May, I attended Tristan's presentation of a 4-year study to identify a possibly new species of agave. This was a great presentation of a true adventure and included information that Tristan had used iNaturalist in his research. And this sparked my interest because I've been participating in bio blitzes using iNaturalist now for what, 4 or 5 years. And this is the first time I've ever heard of anybody actually, well, I knew people used this data in their research, but this is the first time I actually met someone who used iNaturalist in their research. So I called Tristan up and we connected, and here we are tonight. And Tristan's going to present the story of his 4-year study, and in a topic entitled Shining a Light on a Hidden Agave, the Power of Limitations of iNaturalist Records. So get your questions ready. We're going to have Tristan's presentation, and then we'll have the questions and answers at the end of his presentation. So sit back and enjoy an adventure. It's going to start out with a photo, take us through some gardens, and into Mexico. Tristan, you're on.
Thank you for that.
Thanks for joining us tonight. Let's have a round of applause for Tristan.
Thank you for that introduction. Boy, you went far back into the annals of my life to get a lot of that stuff. But but yeah, suffice it to say, I started out as interested in birds, but when I did move to Arizona about 21 years ago, is when I kind of transitioned to arid environments and got really interested in cactus and succulents. So that's my one of my primary passions and research topics nowadays. And so the story I'm going to tell you tonight is, like I said, it's from a presentation I originally did to the Tucson Club, but I kind of tweaked it for you guys to tell the story about how we came about discovering that there was something different going on and how we use iNaturalist records. And some of the things that the cool things you can actually do with iNaturalist records, and the serious limitations that there are. So there has to be a caveat to everything, of course, right? So yeah, so let's kind of get started. And I've got to get my keyboard in the right place here. So come on. There we go. So to start the story, I will give you a quick background in this group of agaves. Agaves is a pretty large family, restricted to the New World, found all the way from central the United States down into the Andes of South America. The primary radiation for the genus is Mexico. Mexico has well over 200 species of agave, and they often have very small ranges, and conservation-wise are often in much peril. And the group that I'm going to be talking about today is a group of small to medium-sized agaves, which are typically generally identified by leaves that are pretty thin. They don't have those sharp horns on the sides of the leaves that many of the ones that you see in the Arizona area have. So they're nice to have in the landscape, a little bit easier to handle and manage. But to give you some idea of the research explosion in this group, back in 1982, Gentry, who was the leading agave expert back in those days, who did live in Arizona and worked in Tucson, produced a compendium of the agave genus within the North American continent, including Mexico. And at the time, there were 3 species in this section, and the section is called Jensenei, so it's a level down from family. And that was agave striata, agave stricta, and agave dealbata, which back in those days was called Dasyleroides, but taxonomy being that what it is, things have changed, and we found out the actual correct name is dealbata. But since 1982, there's been a virtual explosion of discoveries of additional members of this group. Probably the most famous of them all, if anybody knows anything about agaves, is agave pilosa, which was described in 2007, which is a real cool agave. It's kind of small, no spines to speak of. It does have a spine at the tip of the leaves, but the neat thing is that the fibers of the leaf on the distal, like 1/4 inch of each leaf, have frayed, and they turn into like white tufts. So the tips of the leaves look like white tufts of cotton. So very unique. So that was a big discovery, and you see on eBay for hundreds of dollars. They're very hard to get a hold of. But in addition to that, there were 7 other species that were described, and all the way up until 2019 is the latest one. So it's been a very active area of speciation evolutionarily, and clearly an active area of study for botanists that are traveling in Mexico. And no study with agave can be done without Mr. Agave himself. Anybody that knows anything about agaves in Mexico probably has heard of Mr. Agave, otherwise known as Greg Starr. He does live in Tucson, and he goes and he does talks all the time. And he's discovered species of agaves, and he and I got together several years ago, and he knew that I did a lot of expeditions for birds in South America and places, and so he does a lot of exploratory botanical expeditions for agave studies. So he kind of wanted a partner in crime with some background in science. So he invited me to go on one of his botany expeditions for agaves, and the rest is kind of history. So we've been going at least once a year. We've been traveling down there, and we've already started describing several different species of agaves. We're currently working on 2 of them right now. So it's a very lucrative relationship for the 2 of us, and we really enjoy each other's company. Greg's a really great guy. If you've ever met him, he's got a great sense of humor. But the group I'm talking about is the agave striata group, and there are 2 known subspecies of agave striata, or named subspecies, I should say. The nominate subspecies, Striata striata, and the falcata, agave striata falcata. Agave striata is very thin-leaved. It is very cespitose, so it pups a lot. So you see these masses or clumps of the agave because they offset, and you get multiple individual plantlets. They're identical clones of the original parent, but very distinctive the way that they grow. Falcata grows the same way, so it offsets, but as you can see, the leaves are what's called falcate, which is kind of saber-shaped and curved, so you can easily distinguish falcata from nominate striata. And the reason we were studying this group is because back about 7 years ago, Greg Starr was on a botanical survey expedition for a CSSA tour, the Cactus and Succulent Society of America tour. He was going to lead with Jeff Chemnick, and who everybody may know is does some tours in Mexico. And they came across this agave that looked like striata for the most part in the mountains just south of Monterrey and Saltillo in northeastern Mexico. The plants were not flowering. And for anybody that knows anything about agave taxonomy and classification, you really need to have flowers to tell the difference between a lot of these species, because without the flowers, they're very similar. As a matter of fact, agave stricta and agave striata, one of the main ways to tell them apart is actually a ratio of flower measurements. So that's how critical flowers can be in this group to distinguish different species from each other. So he didn't know if it was 100% striata or not. It was interesting. So he was planning a trip to go down there, and he gave me a call and said, Tristan, I'm planning a trip to Northeast Mexico. I want to try to get this interesting agave striata-like plant in flower to see if we can figure out, is this just another variable population of striata, or what do we have here? And for those who are not familiar with taxonomical indicators, CF before the species name is a Latin abbreviation that basically means could be or kind of, it may be. So it's not 100% striata, it may be striata, but it might be something different, but we think it's related to striata. So of course, you know, I'm easy. So I said, sure, sign me up. Definitely would love to do that. So we started planning the trip. And so here's a map of Mexico to give you an idea of where we were going. That's where we were going. So that little red dot in the yellow square is Monterrey. This state here is Nuevo León, and you've got Tamaulipas here and San Luis Potosi to kind of give you an idea of where we are. Obviously, there's Texas, the Panhandle, Big Bend National Park there, and Arizona is way up here. And like the crazies that we are, we drove. So we were planning this trip, and so it's a pretty big planning endeavor to make sure that we got everything set up ahead of time. And while we were planning this, my sister, who lives about 5 miles from me, I live up in Chandler, and she had gotten into agaves pretty big when she moved in to Arizona back in 2012. And sibling rivalry being what it is, I immediately started getting into agaves too. So we had this kind of competition, and basically she's winning because all of hers like to flower for her, and I've had them for years and years, and some of them never flowered. Well, sure enough, she had a flowering agave right about this time that we were planning this trip. And the agave is called Agave tenuifolia. That was a group of that Jensenate on my first slide that was described new to science in 1995. So it's one of the older ones, but it was after that original monograph that was written in 1982. So 1995, Agave tenuifolia was described from Querétaro, which is down in central Mexico, in the mountains in central Mexico, all the way up to northern Tamaulipas and near the Monterrey area. And when I started looking through the literature, because I'm a Virgo, so I have to research everything with regards to tenuifolia, I remembered that 2 years before this, I had actually bought a couple of agaves from a now defunct nursery called Yucca-Do Nursery in Texas. And Carl and Wade were the owners of that nursery at the time. And I remembered there was something weird about the tenuifolia that I got from them. So I went back to my notes, which of course I have a spreadsheet of all of my stuff. Like a good Virgo, I don't go to the shed without a spreadsheet. And sure enough, I pulled back my notes on it and the photo that they published on their website where I bought the plant from. And in their notes, they said something strange is going on with tenuifolia, because we actually were in Mexico collecting seed for our nursery years ago, collected seed in the southern portion in Querétaro, and we collected seed from a population up in Nuevo León, near the Tamaulipas population that was in the original description in 1995. We grew them out, same age, and lo and behold, one population flowered like this. So you can see the flowers are all concentrated at the tip. Whereas if you look at the one in my sister's yard, you see the flowers are in single and double pairs all the way up for almost half of the entire stalk. So they said that's what they saw in the differences between the two populations. They said, something's going on. We have no clue what's going on, just FYI. So like a good Virgo, I bought that plant and got one, and it was actually growing it in my yard. Had not flowered yet, so I didn't have direct comparative material. So I was really interested, and it's like, well, their locality was actually, this is the southern locality, the Querétaro population for tenuifolia, where they collected the population that flowered like my sisters did. And then here's where the population that they collected seed of the plant that had all the flowers congested towards the tip. So that's a very interesting distribution. And the record from the original description of tenuifolia from 1995 was up here. So it was natural to assume that these were also tenuifolia, and that it just had this distribution along the eastern edge of the Cordillera here, because there's a mountain range that kind of goes along the eastern edge of Mexico right here. So when I brought this up to Greg Starr, I mentioned to him the weird thing that was going on. There's something happening with tenuifolia, which is related to striata and what we were working on. So maybe we would look into that as well. And I looked in, at the place that we're going and compared it to where Greg said he actually had observed tenuifolia years before as well. And interestingly enough, he found his old photos, and his tenuifolia all has the congested flowers. So that's not what the original description is. The original description has flowers like my sister's plant had. So it's like, well, where did you see this one? And guess where he saw it? He saw it right in that northern edge of their range again. So now we started seeing some consistency in what we're finding. The northern plants seem to always have this congested flowers, whereas the southern plants seem to have the separated flowers. So we knew something weird is going on. So it's like, let's look a little bit further in this. So what I did is I went to iNaturalist. iNaturalist, as you all know, is sort of a crowdfunding type of website where anybody can go in, set up an account, and you can contribute your observations of any type of organism to this massive international database. It can be birds, it can be insects, it could be plants, it could be slugs, almost anything that's alive. You can enter your own observations. The more data you submit, the better the observation is. So I went to iNaturalist, like, where are all those records of agave tenuifolia? And this is what it comes back with. So they're scattered in this range, but there's the range of agave tenuifolia records according to iNaturalist. And it's like, okay, that fits the range of the distribution originally published as part of the original description in 1995, all the way in Nuevo León and Tamaulipas through Querétaro, which is that pink state here in the south. It's like, well, that fits. But then if I go a little more granular and look at patterns within that overall distribution, this is what I find. There's actually a cluster of observations here, then there's a gap, and then there's another cluster of observations there. So it wasn't a continuous distribution of observations. There was actually a gap in there. And so I went to all of the records with photos, because you can enter a record, but you don't have to put a photo in there, right? But they're much better with photos. And of course, all of the agaves in the group, like I said, are so close to each other that they are effectively identical unless we have flowers. And the differences I was seeing was based upon the flower stock. So what I did is I went to all of the records that had photos on iNaturalist for agave tenuifolia and looked at the photos and determined whether I could see the flower stalk in the photos. So if I couldn't see the flower stalk, I just kind of tossed the record aside, can't decide. But if I saw a photo and it had the flower stalk, it could be the old flower stalk with the fruits on it. I could still tell the positioning of the original flowers if I look at most of them, or whether it was flowered or anything. And sure enough, this is the pattern I was seeing. In that northern population, these are the types of photos I was seeing. You can see up at the top, all of the flowers were clustered at the top of that inflorescence, that flowering stalk, for these records in this northern population. But in the southern population, when you saw photos, you're getting the flowers clearly separated in these 2Z pairs. So quite different from what you're seeing in the north. And according to the original description of Agave tenuifolia, this is what the way that the flower stalks were described back in 1995. So we knew, okay, we're onto something. But it's in iNaturalist, right? Everybody's got access to iNaturalist. So it's basically a race to figure this out, because we're thinking there might be a hidden species, a hidden undescribed species in all of this data that's out there to the world. And of course, if you know anything about taxonomy and describing new species, whoever describes it first, they get all the credit. That name sticks. Rules of nomenclature, that sticks forever. Doesn't matter whether it was published in a newspaper or a professional journal. So we've got to figure this out fast, because we know the public has access to all this. And all it does is just for taking one guy to figure this, to look at these and figure it out, that they know that something's going on. So we said, okay, we're adding this to our trip. And this was in 2019. And what we wanted to do was at least see if there was consistency in those populations of quote unquote tenuifolia in the north, near where we were doing the striata observation. So this was our route. This is, I don't know if anybody used Gaia before. I use Gaia. It's an app on my phone, which I can turn it on every morning, and it keeps track of my day, and each day is a different color. Any photo I take along the way with my phone is automatically added with GPS coordinates. So it's really a wonderful program. So you can see we took a whole day to drive down to here, and then another day to do this. So this area up here is where we were doing the research on striata, that weird striata that Greg had found several years before. We were looking at all the populations of striata and getting some data here. This southern portion here is where we went into the range of that northern tenuifolia population that he had seen and taken a picture of years ago and had that congested inflorescence. So this is, to give you an idea, here's Tampico right here, because I'll show you what subsequent trips, you know, how far they went down, but to give you an idea. So we're north of Tampico, and this is basically where we were researching the tenuifolia populations, and then the rest of it was the striata populations. So really for tenuifolia, like I said, we were looking for some consistency in the distinguishing characteristics that we were seeing, to make sure that it wasn't just a fluke that we were dealing with. On the way, we stopped for pretty plants and pretty birds. I actually got Greg into birds now, so we stopped for lots of birds along the way. And so here's Agave lechuguilla, which is pretty common in the Chihuahuan Desert. And we also went through a mountain pass, pretty cool agave that's there is Agave bracteosa, also called the squid agave. Pretty cool plant. And since I'm not 100% an agave guy, I'm also a cactus guy. That's my actual primary interest in cactus and succulents is the cactus. We had to stop for cacti whenever we could, so this is a nice one. But we did eventually reach our destination. Our first destination was to check out the populations of Agave striata that Greg had found several years before, and was wondering if the flowers make it different enough from regular striata. And lo and behold, we found the plants, but they're not quite in flower. So as you can see, the inflorescences are coming in, but they're not all the way out. We looked and looked and looked, and we found some that were in fruit. So, but this is an in fruit one, so it's past time for flowering. And this was in June. And so we figured, well, we got here too late. All right. We're not going to find the flowers of this guy. We're going to have to come back at some point. We're going to have to come earlier in the year. And sometimes it's a crapshoot trying to guess, not only, you know, from one year to the next, but because of a lot of the climate changes that are happening, things don't go in cycles like they used to. So even if it may have bloomed earlier or later in the past 10 years, you know, some years they're just going to have different variables that are impacting when they're available for flower. So it's just part of the whole process. So for that part of the project, for the striata part, we were kind of stuck. It's like, well, we're going to have to wait for another year and come a little bit earlier. So we moved on a little bit south to check out the locality where he had that tenuifolia in the northern part of the range that he took pictures of the flowers, and the flowers were congested. Oh, and we did check a couple of other populations of striata. You can see those are dazzlerians in the background, but you can see here's clumps of striata that we were actually researching. And when we got closer, sure enough, they were always past flowering as well. So we knew we had to come back earlier for the striata components. So, so we went to what we call Loma Alta, and that's where Greg had found tenuifolia, what he was calling tenuifolia before, and had that photo that was, interestingly enough, congested inflorescence. And it was down in gravel beds of this stream. So luckily he bought this brand new 4-wheel drive truck a month before we left, so we made it and found the plants. Sure enough, there they are. This is what everybody's been calling tenuifolia, right? So it's CF tenuifolia. We were looking for flowers. We didn't find any flowers. So we're too early or too late again for them. However, we did find infructescences, and inflorescence is a flower stalk, and infructescence is a fruit stalk. So it's the same stalk, it's just after the fruits have formed. And you can see in the infructescence up here that all of the fruits are congested at the top, which implies, of course, that's where the flowers were too, right? So now that we saw that, and we saw it consistently, there's him finding another one in this cool kind of habitat. Again, all the fruits are in the distal end. So we know we're onto something now. It's a very consistent difference. We're never seeing scattered flowers or scattered fruits along a stalk. We're always seeing the inflorescences with the same morphology. So we're pretty sure know what's going on. Is there's some difference between the northern and the southern populations. So it's pretty exciting. We knew what we did, but the problem is you cannot describe a new species and confirm all these differences without flowers. So we have to come back because we couldn't find the flowers. So we knew we had to do a return trip the following year, just a little bit earlier, probably in May. So we continued and did some more investigations on the striatas, the different populations on the west slope, and we found one on the east side. And then we, of course, had to visit some really cool habitat and cool agaves that we were passing by. And this is one of the coolest out there, Agave montana. It gets these incredible colors when it's stressed, when it's flowering. So they get these wonderful orange and yellow coloration in them. So my screen's frozen a little bit here. There. So that was wonderful to go up to the mountains and see that really quickly. But ultimately, we were there primarily for striata. So we were doing collecting and things like that for his research in case we were to be able to catch those plants in flower the next year. So all of that in our minds, as we're coming back, we're planning the next trip. We got to get right back down there because we know that iNaturalist has all these things out in the public. We know we got to come back in May of the next year, and we know that we want to do the collecting, and we know where to go. We've got GPS coordinates of several localities now, and our main objective would actually be see where that cutoff is. Is iNaturalist representative of the true range of the northern versus the southern populations of what were considered tenuifolia, or is that artificial? It's just sampling bias. So we're going to try to get in places along the range that haven't been sampled before and see if we can figure that out. So we needed to not only find the flowers, but we needed to know how extensive is the range. Where does that break occur? Is the break sudden? Or is there some kind of cline of the flower morphology changes gradually as you go south? So those are the kind of things we knew we needed to ask. So we started planning everything. We started writing grants. And anybody want to guess what happened next? The good old COVID. Yes, that's right about when COVID hit. Everything shut down. Nobody could travel anywhere. Borders were closed. And so we literally had to cool our heels and keep our mouth shut for over a year, waiting for the conditions to change so that we could get back over that border and do this research and hope in the meantime that nobody else has actually figured this out and published. Because there are several Mexicans that are agave specialists as well, and they publish in international journals as well as their internal Mexican journals. So certainly a very, very possible occurrence for this to happen. It just takes one person to kind of figure it out from records that are in the public eye. Well, when finally the borders did open and we were allowed to go across, it was now June of 21. is when we were able to schedule our next trip. So June of 21, we actually it was May of 21. I have it wrong on this slide. It was June of 19 when we went down the first time. And so we went in May of 21 to go earlier to hope to catch the flowers. So you can see once again, down here where Tampico is, to kind of orient you. Remember, we were up northwest of Tampico before, but now our plan is to explore the entire range to find out where that break is, if there is a break. And down here is where the Querétaro specimens would be, the plants would be, which have the separated flowers from what we'd seen. And up here is where all the specimens that we'd seen with congested flowers would occur. And there's the Cordillera, you can see right down the center. And so we were going to do the east side slope and the west side slope, because we were seeing from our striata research that the west had striata for most part, whereas the east had tenuifolia. West side's very much drier. There's a rain shadow effect from the mountains, and the east slope is a bit more wet. Although we did find some specimens of striata out in the lowlands east of the mountain range. So that was our plan. So to figure out which we figured at this point, we were pretty sure this is an undescribed species. It's kind of hidden within tenuifolia and people don't realize they're there. And so our goal was to figure that out, as well as figure out where's tenuifolia, where's the real thing, and how extensive is that, based upon the iNaturalist records that we saw and the literature that was available at the time. The problem is, in 2019, when we couldn't go anywhere, sure enough, the Mexicans published a paper. And they published it in a Mexican journal, but luckily that Mexican journal was peer reviewed, and one of the peers that was assigned to review it was Greg Starr himself. So he got advance notice that, oh, the Mexicans figured something out in here, which could be a problem for us. 2 years ago, Greg got beaten by a couple of Mexican botanists by less than a month. His paper describing a new species was in press when the Mexicans published their version of the paper. Neither of us knew each other was doing that research. And so their name got published first, so had to retract Greg. So Greg was a little nervous about this. So it's like, well, we've got to add checking out the locality for Lexii into our plans. It's right smack dab where we're going anyhow. So we've got to figure it out. So did they figure it out? Did they discover it? From the description, it looked like it was quite different from what we were seeing, but we wanted to make sure, obviously, that we had the correct idea of what was going on, because this one kind of sits in the middle between what we were thinking were the new species and the ones that we were calling tenuifolia, true tenuifolia. So always stuff added to it. Thank you, COVID. So our first stop up in the northern end of the range, we found the plants easily. They were actually fairly common. Here's a nice hillside of them. You can see they're thin-leaved. See any flower stalks? We were so frustrated. It's like, oh, please tell us that we got here in time. We looked and looked. We saw some that were starting. So here's some that are starting, but they're not fully developed. So it's like, oh my gosh, we probably should have come like a couple of weeks later now. But then we got lucky. It's like we found some way up high. It took some acrobatics, to say the least. Luckily, Greg's about 6'2", 6'3", and has long legs. I'm all of 5'4" in heels. So he found it a little bit easier to clamber up the cliffs to go get them. Oh, darn. But yeah, we found flowers on them. And sure enough, not only are the flowers all congested, which is what we predicted based upon what we'd seen, but what we were finding is, interestingly enough, look at the orientation of the flowers. All of the flowers are, almost all of them are pointed. And this is after we had collected the inflorescence and some leaves. We don't collect the whole plants. We just collect the inflorescence and we collect the leaves for the herbarium specimens. It was actually horizontal. So these plants are on the cliffs, as you saw, and the inflorescence kind of extends out horizontally. It hangs out, and all the flowers were pointed straight up. So it's very interesting because we hadn't seen that before in any species of agave in this group. Even striata, when it's kind of on, it's mostly in flat ground, but when it is on some elevations, the stalks still have flowers that are open all the way around the inflorescence. So that we thought that was kind of interesting. So our next venture took us over to the west side. So we went over on the road, the same road where we saw that first specimens and had them flowering of this new species. We drove over the crest, which gets pretty high, and it goes to the west side, which gets pretty dry because there's a rain shadow effect. And we thought that these might be the new species our previous trip. So we wanted to make sure because they didn't quite match habitat wise, but it was very close where the way the crow flies, it was just this difference of habitat based upon being on the west side. We got there and sure enough, these were more classic striata. We confirmed these were not the new species at all. These were indeed striata, that very clumping behavior, the leaf rigidity, leaf length, those types of things. We didn't find these guys flowering, but we did find old inflorescences. But striata and the new species actually have similar inflorescences in that the flowers are congested towards the tip. But we were pretty sure that these west side ones were striata. So that was a good note to add to our research. So we went back to the east side and went to the next locality down. And sure enough, there's that classic bending, drooping inflorescence, and the flowers are all oriented up, straight up again. So very cool difference that we discovered on this trip by finding flowers. So the next place we wanted to check was the same place we had checked the previous trip, where we had seen the infructescences but not flowers, down that old stream bed. And we got there and sure enough, there they were. We had them flowering and they're doing that same thing. It's that very arching inflorescence with the flowers pretty much all oriented straight up. So very cool, very, very distinctive, not found in any other agave that we're familiar with. So we were obviously pretty happy at this point. We're seeing 100% consistency in what we were expecting from our hypothesis as far as what we would find in these areas with these flowers. So we pretty much knew we had a new species, an undescribed species that currently was hidden within another name, Tenuifolia. So now it was just to determine how extensive the range is and where does Tenuifolia kick in. So we had our little picture sessions with our type specimens, which is the specimen that you collect to actually use as the format or of the description that defines that species. And then we moved on south to the locality where that started it all. The old yuccadoo plants that I had ordered from the internet that I got that picture of. They said something's going on. We went to their locality and sure enough, we didn't get flowers on these plants, but there are fresh fruits on it. And you can see they're classic new species. They are congested inflorescences towards the tip of the inflorescences, quite unlike what is true Tenuifolia. So we're very happy about that. We continued on up this road, and this road actually, Wade and Carl had been there so long ago. The entrance to this road is now completely grown over and we could not get to it. We had to actually go way out of our way and around and found another entrance road to get to this road. And it was pretty hairy going, but we wanted to see how high up the plants got. So we continued up and we did find them a couple hundred meters higher in elevation. And you can see here's the long stalk, but the flowers don't start to right here and they're fairly congested. Now the flowers aren't open here, they're in bud. That's why they kind of look a little different. We didn't actually have them open, but they were for the most part oriented straight up once again, like the other ones we had found of that population. So that was pretty exciting. So we confirmed that the ones that Wade and Carl had do indeed belong to this group that's a new species. And so the next stop we made was where the Mexicans had described this agave lexii, that we were wondering, how does that fit into the picture? And is that the same thing that we know this is a new species? So is this the name of what we found? And what we found is these guys are actually really, really different. They're quite different from either Tenuifolia or the new species that we had. It's a completely different habitat. These very calcified rock deposits, very high in elevation. So it's almost pine oak woodland. in the highlands, and the new species that we were working on is in the lower foothills, not in the lowlands, but in the low foothills. So it's quite above where we would see the new species. And also the fruits, although they are sort of congested at the top, they're still fairly well separated. This is the flowers, and the stamens are really long. The flower tubes are a little bit different. They flare open differently. And the plants, look at the plants. The plants are actually really distinctive. They're small. They don't have these very lax flowing leaves, and they seem to have more leaves per the rosette. So we were pretty confident this time. Yeah, Lexii is definitely related to this group, but it's definitely a separate species. It's not something we have to worry about. This is not the same thing as what we were actually finding at all. They're actually kind of a cool blue or bluish green color as well. And this picture, I couldn't figure out when I took it and brought it home, whether this is the orientation or if it was on its side, but it was tough to, I think it's oriented right like this, but it was a pretty tough place to hike around in that crumbling limestone though, where it was sitting. So we were happy about that. So we didn't get preempted by the description of Lexii. And so as we went down the canyon from Lexii, when we left that habitat of Lexii and we started entering the habitat, the more of the drier deciduous forest and with cliffs, we started looking for the new species. When we got into the lower elevations, sure enough, there it was. So another great indicator that these are definitely two species. They're separated by habitat as well as the morphological characters. So that was pretty exciting that we actually found them what we call sympatric. They're not syntopic, so they're not found in exactly the same place, but they're sympatric. They're found in the same general area, but segregate out by one or more other factors. So that was pretty exciting. And sure enough, we saw them, we found the Cryptica, or the new species flowering here. And you're looking at it from below because they were up on the cliffs, and it looks like they're not open, but they're open, but they're all facing up. So you can't, it's hard to see the actual flowers. The stamens are pointing straight up again. So that classic new species orientation of flowers. So we were pretty excited about that. And then we kept going farther south in that gap that I showed you in when I showed you the maps of iNaturalist with the two populations north and south. There actually are two records of tenuifolia in that gap, kind of out in the lowlands. They're kind of out there in the middle of nowhere. And if you look really closely, what they are in is this low foothills, very low extension of the foothills from the north. The habitat is a little bit lower than our typical new species, much lower than the true tenuifolia elevation. And the habitat's kind of a more humid forest as well. So we assumed that those are the new species. We actually tried to get to them. And this is one of the limitations of iNaturalist. iNaturalist can vary how accurate your GPS coordinates are. And just because GPS coordinates are supposed to be able to get within 4 feet, if you don't register a really accurate one with your photo, it may lead you on a wild goose chase. And this is kind of what this did. It's in an actual international biosphere preserve. So it was very, very dense forest where we were, very humid, one of the most humid places I've ever been to. And I've lived in Louisiana before. And we tried desperately to find either of those locations. According to our GPS, we were basically right on top of them, but we did not find them. So we kind of punted on those when we actually finalized the research, but I'll talk a little bit more about that later. So we did try to find the ones in the intervening gap. But as we passed that gap and we got into what was supposedly the true tenuifolia, we didn't find any of these types of agaves until we got higher up into the pine oak woodlands. And sure enough, when we got there, we found true tenuifolia. You'll see there's the stalk, but the flowers start all the way down here and they continue all the way up. The flowers go all the way up. Here's the top flower. So clearly this is similar to what my sister's plant looked like, right? And my sister's plant was indeed from Querétaro, which is where we are right here. These are the plants found in Querétaro that were included in the original description. So these are what we call classic tenuifolia. This is true tenuifolia. This is what their flower stalks look like. So very, very different. So we're very excited to kind of confirm there's this break These guys are found in a different habitat. They're a high elevation. They're quite distinct from what we were seeing up north for this species that we were confirming now that was new and undescribed. So we did our collection, and here's a close-up of one. Actually had flowers a little closer together, still pretty separated, but upon further inspection, the flowers themselves were very different from all of the other members of the complex, in that the stamens barely exuded from the actual tepal tips of these very flowers. The petals and the tepals were not reflexed, so they weren't spread out like a typical flower. They're very closed like that, and they're barely coming out of the tubes. And so that was a very interesting character that we found that wasn't included in the original description, and it distinguishes Tenuifolia from all the other members of the group. So we were pretty excited. We had all of the data we needed. We had flowers from all the different intervening populations. We did want to check out one other locality way up north to get the farthest north record that we could possibly find, which was just right outside of Monterrey, Mexico. And sure enough, we found them. And of course, the northern populations we would expect to be the undescribed species with the congested inflorescences. And we sure enough found them with flowers. And sure enough, you can see all the flowers congested at the tips. And what direction are they pointing? They're all pointing straight up, just like the classic ones we were seeing. So we were all thumbs up. That was our last locality where we collected all the specimens and did all the legwork, and we were ready to go back to the States and get this written up and published and out there to the world. Before doing that, we have to actually drop off all the specimens, and Greg Starr's on the right, guilty party me on the left, and our Mexican botanist counterpart that we worked with, he's made sure we had all the right permits. That's where the majority of the specimens are going to reside, is in the herbarium in Saltillo, which is where he's a professor, Jose Angel. Anybody knows about the whale tongue agave they might have heard of? He was part of the discovery and description of the whale tongue agave abatifolia. So he was instrumental in getting all this done and made sure we did everything legally out there. And we turned the specimens over to him to do the hard work, like put him in their plant presses and drying them and getting them all ready to do the research. So we headed back and we worked on it. And in November of 2021, out of the Cactus and Succulent Journal for the Cactus and Succulent Society of North America, we published a cryptic new species of agave and an amplified description of agave tenuifolia. Because what we found is we had to redescribe tenuifolia, because tenuifolia was inadvertently described by concluding specimens of a different species, this undescribed species. When they described tenuifolia, the specimens that they used to describe it were all from Querétaro, the southern population, but they had one specimen from the northern population, but that specimen didn't have a flower stalk. And so they just had the leaves. So they just assumed, because the leaves looked the same, that it was the same species. And so the description included measurements of that specimen. So we had to re-describe tenuifolia and make sure it excluded all of the ones that we know now were different species. And we called that species agave cryptica. And a cryptic species is a real thing. A cryptic species is defined as a species that has been hidden, hence the name cryptic, within the name of another species or species. And so we thought this was a perfect name for this plant because it was hidden not just in tenuifolia under that name, but we actually found out that Gentry himself, when he published his monograph in 1982, included 2 specimens of what ends up being cryptica in his write-up of agave striata. So that's how similar all these guys had been and what confusion it had caused all these years. And so, as you can see, this story, a lot of it hinged on and was based upon iNaturalist records. So I want to talk a little bit about iNaturalist records and how they worked with us and how such of a great resource that they are, that you need to actually be cognizant of the limitations as well. And that's extremely important. So per iNaturalist, the data quality assessment is done on all of the records, and it's a summary of an observation's accuracy, a completeness, and suitability for sharing that data, right? Because we know that anybody can go out and put anything on there. So there is some type of administration and rules behind the quality of a data record. So it has to be, in order to be a verifiable observation, it has to be an observation that has a date, is georeferenced. And like I said, that's kind of a bit of a problem because different accuracies of latitude and longitude can be input, and there's no way to register that, right? It has photos or sounds, because a lot of them do not have photos. And it isn't of captive or cultivated or origin, which sometimes even with tenuifolia is a problem, because we know that there's one record of tenuifolia out there that's from a garden near Mexico City. Totally out of range. We knew it was a problem to begin with. But we don't use all of these records because of the variability in the accuracy of these records. There is a grading system that iNaturalist has. And in order to be research grade, the community, in other words, anybody that has an ID, that's part of the difficulty of defining the community. Anybody that's got an ID can agree on the species level ID or lower, so subspecies or variety, when more than 2/3 of the identifiers agree on a taxon, which seems like a great idea, right? Well, 2/3 of the people say it's this, only 1/3 say it's something else. So I'm going to consider it research grade for the one that 2/3 of the community agree on. And the reason that is important is there are global databases that utilize iNaturalist data, and iNaturalist sends data feeds to these databases regularly of their quote unquote research grade records. They don't send records that are considered not research grade. So that's where this qualification comes into play. So when we published our paper, we didn't cite iNaturalist specifically. Well, we did in a couple of places. We mentioned how we used iNaturalist, but we used the database that iNaturalist populates, because there's other things. There's specimen records from museums, and there's other databases that come all together in a single, and it's called GBIF database that we used. And the nice thing about that is, excuse me, when I do my selection of data from the GBIF database, it actually saves that selection so that it's repeatable. Science it's important to do repeatable research, right? So anybody can actually go out there and see what data I looked at. They know exactly, it's saved out there for perpetuity. And there's a link to it, as a matter of fact, in the literature cited section of the paper. So it's a DOI link to that data set, so anybody can see what we were looking at. So what we had to do is we couldn't just use the dump of research grade, because we really had to be sure what we were looking at. So I went back, and once I did the dump of the research grade data from iNaturalist and the other sources, I actually went through each one by one and only included them if there was a verifiable specimen or photo with an inflorescence, so with the flower stalk, so that I knew 100% to categorize it as one tenuifolia or the new species. So a lot of work had to be done to make sure that the iNaturalist records could be used. And that's the important lesson here. They're not to be used out of the box for a rigorous scientific purpose. You really have to be careful if you understand the rules in iNaturalist. And one of the illustrations of this can be this plant right here. So right now, this is a plant. I just took this off of iNaturalist this week, okay? So this is Agave tenuifolia, right? And Lex Garcia is the guy who actually put this out here back in October of 2015. Now, you might recognize the name Lex Garcia. Agave lexii was that new one. Sure enough, he's one of the guys that described the new species Agave lexii in 2019. So he knows this stuff, right? And I've actually been communicating with him this whole time. He was actually very instrumental in some of the components of this, and is acknowledged in our paper as such. But this record is out there right now as Agave tenuifolia. Why is that? This is the record. This is a photo. If I go out and search for research grade records of Agave tenuifolia, this is what I get. Red squares. Look at all these red squares up in the north. And we just talked about these red squares up in the north should not be tenuifolia. They should be down here, this is tenuifolia down here. This rectangle here is actually 2 orange squares. Those are the 2 records that we tried to find and we couldn't locate them. Okay, so they're still recorded as tenuifolia as well, but they're research grade. So these are ones that are being passed through the databases, right? But I went in to iNaturalist when this published and I put in there, this is actually Cryptica and here's the reason. But because in October of 15 and January of 17, we've got now 1, 2, 3 out of 4 IDs still say tenuifolia. It's still identified as tenuifolia in the research records. So it's really critical that we understand and look at these individual records individually and assess whether or not they're appropriate to include in the research. We have to be very, very careful because just doing a general search like this is going to give somebody a very incorrect idea of the distribution of tenuifolia. And if it's used for another scientific assumption like conservation purposes, land conservation or things like that, then of course we're going to have big problems in the future, right? So that's why I want to call out that although iNaturalist absolutely is a huge, huge help, like any crowdsourced type of database, you have to be very careful about the data points that you use within it and be very selective and understand what you can and cannot use. Now, that given, it was, like I said, very helpful, and we did some really cool analyses with them. One thing we did is we did a plot. We mapped out the research GF, these are the GBIF records, so it includes iNaturalist records and the other records in that GBIF database that I was telling you about, that we verified with pictures or specimen records of the inflorescence, so we know how to assign these. We mapped the distribution. So you can see the yellow is Cryptica. That's the new one. Then there's this big gap, and then the green is tenuifolia, way up higher in the mountains. So really very clear picture of the distribution now. The interesting thing is the red represents striata. Striata is west slope, and literally you go right over the ridge to the west slope and poof, they change to striata. So a very distinct habitat preference for these species as well. Here's Lexii right here, the blue one. That's the one of 2 localities that are right next to each other of Lexii. And we have the lower data points down lower that we found them down lower in the canyon. So different habitats. So that's what we basically call effectively sympatric species. So that was really interesting that we could include those. With specimen records are the ones that are surrounded by the deep black, and the visual records are the GBIF records are the small triangles. So really helpful in where iNaturalist verified records by us paints a really clear picture of what's happening. The 2 unknown ones that we tried to get to are in this range of hills right here. And if you look at the map, it kind of makes sense that they would be Cryptica. So we expect these, when we do eventually find them, to be Cryptica. It's a natural extension of that habitat in that range. It's very clear when we got back and looked at the detailed maps. So that's what we suspect. And tenuifolia, even though it's in between tenuifolia and Cryptica, tenuifolia is much different habitat, and that range would more likely be Cryptica. So that's what we said in the paper. Those are likely the Cryptica. Another cool thing we did is I have a friend who works with the IUCN for cactus conservation, and he's working on his PhD, and he's developed some modeling techniques of modeling habitat usage by cacti and how it's affected, how global change is actually affecting the distribution of cacti. Because everybody thinks, oh, cacti are going to be, happy that we've got this global warming, right? Because cacti love heat and everything. And what he's finding is the opposite is true. And he just published a major paper that demonstrated that actual global warming is really, really bad for cacti as well. But because he had this model, we started looking at our data, and he said, I could use those data points, input it into my program, and come up with a probability scoring process to give you a probability of the distribution. Because as you see in the map, there's some isolated hills out here. Up here, there's a lot of ridges that seem to be similar in height and perhaps habitat. And so we actually plugged the iNaturalist records, the GBIF records that were verified into his modeling program. And we actually came up with a really cool way of demonstrating the possible distribution of the new species. So these are agave cryptica records that are compared to habitat variables in the range of cryptica. And so it comes up with a probability, and the darker green is the higher probability. And sure enough, it's the east slope of the Cordillera. They're more highly probable, right? This is where that southernmost data point that we had was of cryptica. And then here are the extensions where the two anomalies, what we couldn't find, are down here. So they're actually within the probability of distribution based upon his modeling. But really interesting is, sure enough, there's some probability that there's some good habitat out here in these isolated hills that we're going to check on our next trip to extend the distribution, as well as lots of really good habitat north of Monterey, because Monterey is all the way down here. And actually, Greg found some old photos from agave he took a picture of like up here years ago. And sure enough, it shows a cryptica-like inflorescence. He just has a partial picture of flowering stalk, and sure enough. So the range of cryptica probably extends well above up in this areas, which is, it matches iNaturalist for the non-photo flower stalks, the ones we ignored. So getting those to change to tenuifolia records is going to take a long time, but I've been trying to update iNaturalist with the new ID, but it takes 2/3 of people to agree with me before it actually changes on the database. So that was really neat and a great way we were able to use iNaturalist records for some of these modern techniques. So when you looked at this is going back to the original distribution that I looked at in iNaturalist when I first was starting to look at this, that was the oval. I did lie a little bit, not really lie, but I left something out. There were records all within this rectangle, but there were actually a couple of anomalous records all the way down here, which didn't make any sense at all, because this Cordillera kind of stops here, becomes a massive volcanic range of high mountains, and the high mountains come down here. This is all very humid here, and the mountains are humid, but there's like rain shadow effects. And in here, there's like a rain shadow valley. There's a high volcano right here. The moisture coming in from the Gulf makes it very dry. And there's a couple of low karst hills down here. And there were a couple of records of agave tenuifolia on those hills. They did have pictures. They had pictures of the fruits in frutescens, no flowers. The infrutescens did indeed look like tenuifolia, in that the fruits were highly scattered along the stalk, but the plants did not look anything like tenuifolia. So we knew just looking at it, this is really not tenuifolia. We couldn't contact the person that put those records out there, so we had no way of verifying. So in the paper, we kind of punted on those, saying these are out there, these are identified as tenuifolia. We do not think they belong to a tenuifolia at all, and we need to be confirmed to be sure. They don't fit the range, the habitat, the morphology that we can see in the pictures or anything. They just need to verify. So that's the way we left it when we submitted the paper. And we submitted the paper in September, and it was eventually published in November. And in October, Greg and I actually did another trip, and this time we went to Oaxaca to work on another undescribed agave that we know is down there. So this is Oaxaca down here in this large brown state. And as we were tooling around there, it was like, we're not that far from where these records of this supposed tenuifolia are. Maybe we can take an extra day and take a jog out there. So we did. We found the plants. They were not flowering, but it was obvious we were correct. They are 100% not agave tenuifolia at all. So it's like, because this is while the paper was in press, right? It was being published at the time. But what we did find out, they weren't even agaves. Not only were they not tenuifolia, but they weren't agaves. So that, my friends, is the next chapter of the story. We actually went back to try to get flowers in March of this year, and they weren't flowering. And unfortunately, we think that because of global warming, and it's in this rain shadow, and the precipitation has been all over the place, is that they don't flower every year now. We found old infracrescences from 2 years ago, but we found nothing from last year and nothing even remotely interested in flowering this year. So we're currently writing grants to actually see if somebody in the nearby university in Mexico would go and check every now and then to see when flowering occurs, because we were there at the ideal flowering time. Everything else was flowering, but not this plant. But it's a very exciting discovery because it is not an agave, but it is related to agaves. And so I hope to be able to tell that story in the next year or two tops. So, and to finish, that's our Cryptica with our classic flowers. That the best identification character on the plant is all that sky pointing flowers. And that is it. So I thank you, and I'm certainly welcome to answer or try to answer any questions you might have.
Very good, Tristan. Very good. So, any questions out there?
I have a question.
Yes.
Because this seems like a race with time, and of course you had your hurdles. Is there a monetary value that you get, or the supporters of the grant that provided the funding, or is it just the recognition?
It's for the benefit of science.
Very good.
Yeah.
I'm
Yeah.
glad that answer.
There are a lot of people that benefit from these types of trips, and much of the grants money is applied to guides, Mexican permitting agencies, all the official stuff. So a lot of people are getting paid, but we aren't. So yeah, so the grants usually cover, we hope, cover most of our transportation and lodging. Of course, some of these places, lodging consists of a tent that we bring down, or a hole in the wall. But yeah, the expenses aren't, there's no salary towards them. For some journals, luckily the CSSA journal doesn't have a charge for publishing, but unfortunately the trend in science now for technical publications is to have a cost associated and that the authors must pay to publish scientific research, which is, I think, abysmal and horrible. But when I discovered a new species of cactus back in 2017, in order to get it published in a respected journal, part of the grant money went to actually paying the fees to get it published in that journal. And I also am a big believer in open publishing. In other words, nowadays, people can publish to the internet. There's these online journals that you can publish to. But if it's not open published, legally, if there's copyright infringement, if you share it publicly outside of author copies. But if you open publish something, that means it is actually open and can be accessed by anybody without any kind of fee or restriction. And in order to open publish something, most of journals will charge you an extra fee to open publish it. So like, extra, and it's not, at least for scientists, it's not a small amount of money. It's $700 to $1,200 typically to open publish a single paper. So a lot of the grants nowadays are trying to offset some of the costs that the scientists have just to get the data published. So unfortunately, stipends are not the usual. Most scientists are associated with a museum or university or a herbarium, and so they are financed, by their salary. But people like myself and Greg Starr, who I have a real job. I work with the computer all day. That I don't have the capacity for that. So that's why we rely heavily on grants. And the TCSS, the one I gave the original talk to, Tucson Cactus Succulent Society, and the Central Arizona Cactus Succulent Society, both of them contributed funding to accomplish this because it was such a long-term project.
Thank you. Could you turn
Yeah, so
off your
Tristan-
screen so we could see
yeah.
everybody?
Sure.
So Tristan, am I right that this, what you showed us was over a 4-year period? Is that right? Of course, there was the COVID break.
Yeah. It started in 2018 is when we started the prep for the trip, when Greg first contacted me about going back to this agave striata that we now know is actually just a variation of striata itself, and so it's not anything distinctive and new. But yeah, that started in 2018 when we first started looking into it, when my sister's agave bloomed, which caused me to go do some research and started us down that path. So our first trip was in 2019, June of 2019, and then COVID happened, shut everything down. 20 was out of the question. Beginning of 21 was out of the question until we finally made plans and returned in May of 21. And then after that trip, we came back and did the writing and the research, and so the publication was in November of 21. So 18 to 21. So like a 3 to 4 years. Yeah.
Mhm. So Tristan, a couple people have asked how you got to these places described by the GPS. Did you went on a paved road, then you went on a dirt road, and then you whatever? How did you get to that point?
Yeah, we used Google Earth, and we plot the GPS coordinates from the records into Google Earth, and we zoom in as much as we can to see if there are any road-like things or trail-like things in the photos, and make a best guess and get down there and ask natives, the locals, how to get to places, or how to, we want to get over here, and are there roads? A lot of roads are private, so they look like we can get there on the Google Maps when we get there, and we can't go in, especially in Oaxaca. Oaxaca is kind of managed differently than all the other states in Mexico. It's kind of like working in a Native American reservation in the US. All of the areas are owned by the communities, and so you actually have to have permission from community leaders to go off the main roads, which means highways, not even a side road of a highway. We got told to get off just a paved road. We were bird watching like a half kilometer on off the main highway, and they caught us immediately and said, no, you can't. It doesn't matter if it's paved or not. You got to get back to the main highway. So it's very difficult to work in Oaxaca. The areas in Northeast where we worked a lot easier, but there were still some, places we couldn't get to where we thought we could. The two records that we still haven't gotten to that we tried to hunt down, it was in the middle of this biosphere preserve, so a lot of vegetation, so we couldn't see where the trails were. So we had to kind of guess, and the habitat's pretty much destroyed right up until the border of the preserve at the fence line. And so we saw the roads going through the farmland. So we got permission from the farmers to take those roads right to the edge of the forest where that fence was, which was really, really bad because it was a sandy mud, and we almost didn't get out. It was the kind that sticks to your shoes, and you carry it with you. And but anyhow, we had to bushwhack from there directly towards those GPS coordinates because there were no trails. It's just overgrown, really miserable and humid weather. We ended up finding a track inside, an old track that we followed for a ways to get as close as we could to the GPS coordinates. But yeah, it's just kind of play it by ear and do the best you can. Like I said, in the place that Carl and Wade had given us directions to get there, because Greg's a good friend of them from that original Yucca-Do nursery. They gave us instructions, go on this road and go here. Well, those roads were completely run over. They were impassable. So we had to play by ear, and we had to kind of ask locals if we could get around and all that kind of stuff. So it's not easy. You have to plan a lot of downtime or into these trips. They're not, let's go down there for a couple of days and come back and get everything we need. It doesn't happen that way in reality. So
interesting. So Kim and I can't just take off tomorrow morning in our truck and go to these spots.
You could. You might get stuck some places, but yeah, it's been, there's always exciting things that happen. We were just in the Northeast where you can, bird watch or whatever, and we were off in the Northeast Tamaulipas and on a side road right next to the highway, and a cop comes over and turned on his lights. So we all come back, and we were just looking at cactus, on the side of the road. And he was just warning us is that, you're very near the border with Nuevo Leon, and the communities in the area just aren't real nice about the border situation. And so they're often not nice to anybody walking around, even though it's no trespassing or anything. So he just was recommending that we move along, So we weren't doing anything wrong, but, he was nice enough to let me know, might want to move along. We're now WhatsApp friends, so we talk to each other all the time. Caught from Tamaulipas, but yeah, so always an adventure, always an adventure. I've been doing this stuff, for over 40 years now, 30 years, 35 years, something like that. So I'm kind of
Andre, did we answer your questions? You had a couple there. Did I get those out? Andre, you're on mute. Andrea, you're on mute.
I was wondering if the Cryptica has been noted in South America, similar latitudes and climatic conditions or soil conditions.
Yeah, no, agaves in of themselves, like I said, are the biggest diversities in Mexico. It drops off very drastically when you start hitting Central America. And probably because a lot of the dry areas are now restricted to Mexico, and you get into more humid areas, and you lose the ability for agaves to survive. And there are agaves that have been adapted to more humid environments, but historically and evolutionarily, they're more of arid adapted plants. So they do drop out diversity-wise once you hit Central America. And then in South America, Colombia's got a few, but the only ones that are found from Colombia south is primarily one, or depending on your taxonomy, one or two species. And there's actually some controversy whether the ones that reach Peru and Bolivia are actually natives, or if they were actually domesticates that were brought over from the pre-Inca civilizations, from the Central America civilizations that they traded with along the coast. So there's still some debate as whether there's any true native Bolivian and Peruvian agaves, but if there is, there's only one currently known, Agave cordillerensis. Otherwise, people lump it in with Agave americana, which is the common one in Mexico that a lot of societies have grown for many years.
How do you feel about introduced plants? When you bring back seeds and whatever and plant them in Arizona, or you order plants from other places, do you find out about the pollinators, or are you comparing how the new plant is with the native plants here in Arizona?
I work with a lot of conservation projects, especially cacti. I'm working with a Brazilian cacti conservation project right now, and so I'm growing a lot of these. But for me, I don't think there's too much of a concern with doing any kind of damage to the local populations. Obviously, we don't want to release them out, and have them acclimate and go wild or anything. That's not ideal. As long as you're cultivating in a controlled cultivation type of scenario, I wouldn't worry about it. But for research purposes, when I do pollination, I always am concerned about cross-pollination. That's very, very important to control that. And for right now, I'm working on Brazilian cacti conservation. So I'm getting seeds from a collector in Brazil, all legal and everything. He's trying to salvage a lot of the species that are going extinct because of the habitat destruction right now. And so he's doing collections. He's actually growing them in his own garden, his professional garden, and then hand pollinating to make sure that we have pure seed from plants from the same localities, and then sending the seeds to me. I have all the import permits for it and everything. And then I share those seeds with local growers that I know are really good, that I trust, that control any pollination efforts that they do. So when they do get to flowering age, a lot of these are, I'll be long dead by the time they start flowering, but it's a long-term project to say the least. When they do start flowering, we are controlling all the pollination. So we bag the flowers, for example, to protect pollination attempts by bees or whatever is doing the pollination.
Oh,
We'll always do
interesting.
a controlled pollination and make sure that we actually pollinate only from plants from the same populations, because we
Oh,
can't necessarily say same species, because as you can see from my talk just now, sometimes we think something's the same species, but it really isn't. And we won't find that until later. So that's why it's important when you do cross-pollinate that you really try to cross-pollinate with plants from the same populations, not just the same species. And then, we have a long-term goal of preserving these and distributing them to professional growers. So in April, we just delivered 120 seedlings of Brazilian cacti that are basically unknown in cultivation outside of Brazil to the Huntington Gardens in
oh, nice.
California, so that they can start trying to propagate them for the ISI, to propagate them for posterity as well. And then as we start producing these controlled seed generations, we'll also donate those seeds to the cryo storage at the DBG here in Phoenix, as well as the Huntington, for long-term preservation as well. So, but it's all about controlling the pollination. When you're talking about science and research and long-term viability, it really takes a lot of effort to control the pollination because of the local, butterflies, they don't care, moths do care apparently, because they're very yucca specific, but yeah, you do have to be very careful when you're introducing non-native plants and you're doing some kind of research, anything besides an ornamental type of display.
Oh, thank you. That's very interesting. I have another question. If somebody else doesn't right away, does the botanical garden in Oaxaca have one of the crypticas?
Well, the cryptica is only found in the north, and Oaxaca's in the south. So
But
probably don't.
the garden has, like DBG has plants from all over Mexico.
We haven't visited the garden in Oaxaca. In Oaxaca, we're working with the herbarium and instead of the garden, because we're collecting scientific specimens. So we've been working with Cervo, which is the herbarium in Oaxaca, for our work in Oaxaca.
Okay.
So I haven't visited the botanical garden in Oaxaca, unfortunately. I would love to, but it's always so time schedule tight. You'd love to take a couple of days off or something to do some fun stuff, but the research won't let you.
Thank you. That was very exciting what you're doing.
Oh, well, thank you. It's fun.
Any other questions out there? Okay, so I have one, and this will be our last question. So what advice do you have to us, Tristan, as we go out and do our bio blitzes and we collect data for iNaturalist? For example, what should we try to do with the photo? What should we try to do with the naming? What should we try to do with our input that would help someone like you when they do their research?
Fantastic question. I'm very glad you asked that, because one of the things that I think is grossly underused, and this is typical, it's a spot where you can hurry up and get stuff loaded, right? You come back from a trip and you just load a whole bunch of bird observations, you load a whole bunch of plant observations. There is an opportunity whenever you enter an observation to provide information about how you identified that plant. So how did you actually come up with that ID? Did you use a specific source? You saw when in my entry where I said, this is Cryptica, it's not Tenuifolia. I didn't just say Cryptica, I think this is Cryptica. I gave a justification. It's like, there's been recent research showing that plants in this part of the range are referable to here. Here's the citation of that. So if, birds, it's a lot easier, because I work with birds a lot too, so that's why birds come to mind, because often they're visually very different. But plants, not so. Plants are kind of free with their genes, let's say, a little bit, quite a bit. So there's a lot of hybridization that occurs. There's a lot of very distinctive populations depending on where you are. So my two recommendations is make sure you're using as a great a resolution as GPS coordinates as you possibly can. Don't guess. And if you do guess, please put it in your observation that this is a best guess type of observation. a lot of people will go back to older records and best guess their GPS coordinates, which is what we think happened with this 2 records that we tried to find, is that after the fact, they went back and it's like, oh, I was right about here. Well, that could be 5 miles away when you've got no trail and there's dense vegetation, right? It's basically impossible to get to as far as we know. So yeah, I encourage people to use the observation free format section when you put in your identifications to justify your identification. And it doesn't need to be a long justification, but when you know something especially is rather difficult to identify, just put in there, that this is where you got that information and how accurate that information is. Hope that answered your question.
So there I am, I'm out in the field and I take a picture of something and I'm not sure what I have. And the iNaturalist comes back and says, a suggestion. Do I go with that suggestion? Do I say, here's the unidentified cactus, I don't know anymore, and then I take some pictures, etc.
Yeah, unless you have justification for an ID, never just go with the flow. That's my thing. I'm a natural doubter, and I don't know if that's my science background or the fact that I grew up in a family of 7. But but either way, it's like I always want to know for sure. So I would never just go to something and it's like, well, I don't know what it is. They said it was this, so I'm going to agree with it. It's like, no. If you don't have justification, that you don't have a reasoning behind your decision, then just leave it. If somebody suggests something, don't just say, okay, yeah, that's it. Do your research, and we can, the fact that we have the internet nowadays and we have things like iNaturalist and cactiguide.com, and there's all these resources that we have available to us. It doesn't take that much work to do some basic research, really. And that's what's critical in these types of crowdsourced information places, is the lack of research that people really do when they enter their information there. So.
Very good. Well, Tristan, on behalf of the Master Naturalist, thanks for joining us tonight. Your presentation tonight was just as exciting as the first time I heard it. I appreciate you modifying it to add a little bit more about iNaturalist, because I think that was very interesting, and I think for all the shaking heads I see out there, that everybody else is agreeing with me. But Tristan, thanks again, and thanks again for all you do to help educate the naturalists in our community in that.
All right, you're very welcome. You're very welcome.
A round of applause for Tristan.
All right, we'll talk to you guys later. I'll check
Okay.
you to your meeting.
So Jess, we'll turn it back over to you.
Okay. So I was actually just looking at the attendees, and a majority of us were, if not from cohort 4, affiliated with them somehow. So I was originally going to go through everyone's names, but we all, for the most part, except for maybe 2 people, know everyone here. So to save on some time, since we're running a little bit behind, I just want to say thank you guys to cohort 4. You guys were rock stars. Everyone here from cohort 4 obviously is now a Master Naturalist intern or an established one, since some of you are volunteer phenoms. Sharon. So yeah, with that, I actually want to pass it right over to Kathy to make sure she has enough time to hit the 3 things she wanted to talk about tonight. Kathy,
Can you hear me?
Yeah, we can
Not
hear
very
you.
well.
Oh, hang on a minute. Let me disconnect my earbud. All right, can you hear me now?
That's better.
Okay, hold on. Now I can't hear you. There we go. Sorry, I'm on my phone. All right. So forget what I was gonna talk about. Okay. So the annual meeting, thanks, to everybody that did attend the annual meeting. If you weren't able to attend, and feel free to chime in if I miss anything, went really well. We had several field trip opportunities. I think everybody had a really great time on their field trips, and learned something new. Lorianne just kind of had a brief overview of some things, basically the chapters and sort of what our responsibilities are as a chapter. She highlighted a few new things that are in the works, like some embroidered gear, some hats, and a new logo, or I should say an additional logo. I think it's just in addition to the one we currently have. We had some labs, or not labs, I guess workshops, talks. So there were 2 of those, I think. One was the invasive plant species, which Pam led. And then we had Gabby did a environmental education workshop related to community events. So both of those went really well. Yeah, thanks also to everybody who jumped in to help with a field trip or lab. It was great that we had so many master naturalists leading those. And then the last part of the day, we heard from the Rio Reimagine project, and we should have more information coming out on this soon. If you aren't familiar with it, definitely check out the website. I think it's just Rio Reimagined. Can't remember if it's .com or .org, but I'm sure if you Google it, you can find it. Or maybe somebody can find it and throw it in the chat. But that's a really cool project. I actually had not heard of it before the meeting. So it was really neat to learn more about that project and what they're doing. And they definitely are gonna have a need for master naturalists to be involved. They do things like tree plantings at communities along the Rio Salado. There's a bunch of other like large scale projects in the works. So, stay tuned for that. There should be opportunity to be involved with that. And then, yeah, I don't know. Did I miss anything, guys? Does anybody else want to chime in about the annual meeting and what your thoughts were? It was great to see everybody in person and talk to some people. And so with that, I'm going to tell you, and you're going to hear this probably multiple times over the rest of the year, save the date for January 28th, 2023. That will be the next conference, and she's going to basically be sticking to that. I think it's like the last weekend in January. So it'll always be the last week in January. Put it on your calendar now for 2023, 2024. And they're going to be rotating locations. So we did it in Maricopa this time. They might do it in Tucson next year, or we have a chapter now in Prescott area. And then we also have a chapter in Yuma. I think it's Yuma area. It's on the border. So they're going to rotate it around so that we can get to go see what other chapters are doing, and they have an opportunity to lead field trips and as well. All right. And then what was my next thing? Hang on, to toggle over.
I believe it was what's changed or what's changing.
Oh yeah, some changes just to be on the lookout for. Please make sure you read your emails when we send them. But we are going to have some changes coming up with regards to the Google Drive and how that's going to operate. It'll now be like a shared drive, so it's going to be easier to navigate. We're not totally ready to launch that, but be on the lookout for that email. We're going to be sending a link to a new form for you to request that partners be added to your Volgistics. We have a new form we're going to use for that. What else we have coming out? We have some people serving in new committees. Actually, it was the second thing I was going to talk about, the board. So we have some people serving in new committees and new roles. So there may be information coming out on those. And I'm missing something. Kim, what am I missing?
I'm trying to answer someone's chat question, so I was typing, sorry.
Yeah, some information will be coming out about entering your hours into Volgistics. So after our board meeting next week. We should have some more information on that. So just be on the lookout, read the emails we send. They do have important information, and we, want to make sure everybody stays on the same page. And then, of course, at each chapter meeting, we'll have additional, updates and can answer any questions about those things. And then some of the board changes. Well, actually, there will be some board positions opening up. So be on the lookout for that if you're interested in serving. If you're interested in serving on a committee, we still could use help on some committees. So get with us if you're interested in, particularly we need a communications chair and we need a outreach chair. So if you're interested in either of those, let us know. We did have a change to the board. Kim actually is the vice president now, and Alexis is just taking on the treasurer role. So we did have that change recently. And I think that's all I have. Any questions or anything? No? All right. So I think Kim has an update.
Well, we had a problem with our email, with our MailChimp and our email. If anybody suspects they are not getting emails or anything, let me know. We're trying to solve the problem. We know the problem has gotten smaller. There's only, I think, we're hoping a handful of people that are not getting emails, but please let us know. And any hints you have helps us figure it out. There were multiple things involved in the glitch that was happening before. And I could say also, if you can't get a hold of Kathy, I'm helping and supporting her. So, because she's had so much to do for so long of a time. Let me know. I try to help her. I don't know all the answers, but I try to help.
Okay, well, that was more brief than I anticipated. So I guess at this point, I'll open it up to the boards and committees. I hadn't gotten any updates, but if you guys have any updates that you can think of now that we've kind of opened up the floor, feel free to chime in.
I would just like to thank everybody who volunteered to get a job description done for our associates and partners. I know that was a really big job and we're almost there. I have been away on vacation for 2 weeks and just back. So I'll be working with Deb Petrick to see where we are, but we're down to just a handful of those. So thanks to all of you who chipped in and assisted with that. Much appreciated. And please keep looking out for new partners as you are doing travels and visits and such this summer. So if you think someone would be a good associate or partner, please submit one of those. There is a new form. I notice our old form is still up, so we will get that cleaned up over the next couple of weeks. And meanwhile, just contact me and I will send you a copy of the new form.
Mo isn't here tonight, but and Jess has said something already. We had a class of 30 people and 26 graduated. One is deferring to next session. So I think that's a great number. And we will look forward to working with all of them. They are great, super, and they're bringing in projects and new talents. I'm really excited.
Okay, and then while I'm here,
I would-
actually, all right, go ahead, Sharon.
I would just like to say it didn't go out in the newsletter because I was late getting information on it, but National Pollinator Week is coming up from June 20th to 24th. So there's lots of opportunity for advanced training from partner pollinators. And I put some of it on the calendar and some of it's on the spreadsheet. So have a look at that if you're interested in anything to do with butterflies or bees or There's even a presentation on gardening, culture and equity. So the breadth of things that you can have a look at is pretty widespread.
Jess, you are fast. Sharon, she already put the link in the chat for people.
Kathy's got her hand raised, I think.
I do. I just wanted to let everybody know that the bridge over the 60 in Wickeburg, Hassayampa River Bridge, every night from 7:51 to 8 o'clock, we have a major bat leaving. It's amazing. Taking a bunch of
It is
films
such
of
a
it.
good experience.
It is. And so I'm inviting everyone out to Wickenburg.
And it's at the what bridge?
It's at the bridge that you drive over to get to town. And
Okay.
if you stop at the McDonald's and go down the stairs, at 7:1, you will see them leaving.
That's magical.
It is. Are there
This- it is.
vultures?
I-
Are the vultures still coming through, Kathy, or are they done?
Vultures have pretty much stopped, but we have resident vultures, so you will see vultures still. And we've got a lot of juvenile red-tailed hawk today at my house that was trying to get into the pond. I don't know what he was doing, but and we got a lot of beautiful birds, just amazing things coming up in the Berrien area. So check it out. Nice things going on here in Wickenburg.
Awesome. Thank you for that, Kathy. I'm definitely interested, so I might find my way up there shortly. Did anybody else have anything? Okay, so I had two things that I wanted to mention. One, for everyone's clarity, we ask this every time we start these meetings back up. But for a night like tonight, where we did about an hour and a half of further education and about like 15 to 30 minutes of just the volunteer hour equivalent, how do you want us to delineate that? Do you still want us to do 1 hour of each, or do you want us to specify relative to what we're actually accomplishing during the meetings?
Just, just for consistency. I mean, if everybody's cool with it, let's just do an hour of each and just stick with that. It's probably easier, but either way, I don't really care, honestly.
Okay, all right.
But yeah, I think staying with an hour of each is just going to be easier. But yeah, like if you're really hurting for AT and you need that extra half an hour. But I don't think anybody here is hurting for AT, so.
Okay, and then the secondary thing is I actually won't be here for the next meeting. I'll be in Mexico, oddly enough, not doing anything nearly as cool as finding a new species. But if somebody wouldn't mind taking over my duties for the next meeting, that would be greatly appreciated. And I can also remind people closer to the date to see if somebody who isn't present for this meeting might be able to do that. But I wanted to put it out there now before I forget for 3 weeks. And I just, oh, by the way, I won't be here.
Thanks for letting us know, Jess.
No problem. I realized it today. Sorry about that.
And with the AT question Jess just posed, so you'll log because I think a lot of the people in here are from cohort 4. So it gets logged as, it's just AT online. And then volunteer, if you don't have it assigned, let me know. I think everybody should. There's one that's just called like general meeting. It says general, like chapter meetings. That's the one that you'll log the volunteer hours under. If you don't see that, just let me know. And I think that was it.
Kathy?
Yeah.
I just wanted to say that Juanita is short on volunteers. And she is encouraging everyone to volunteer to help out with Maricopa Parks.
All right. Yeah, I mean, I guess I know there's some stuff on the calendar. So all the remaining Maricopa events should be posted on the calendar if people want to go check them out. If you get with Karen, you guys can maybe send an email with if there's some specific things that she needs. But yeah, so if anybody's interested, I guess go look at the calendar or
Yeah.
reach out to you.
Not necessarily me, just reach out to Maricopa County Parks and
Okay.
Juanita. She's been on vacation. I think
Yeah.
she just got back.
Yeah, today.
Yeah, you can always go to the Maricopa website and go to the like volunteer section too, and anything upcoming should show there as well.
And Beth and I are going to implement our midterm project with Juanita. We probably won't be starting until either December or next year, but we'll be wanting to have some research grade phenology trail or phenology walk and phenology type of information for the post-fire assessments on more plants than had been started. So anyway, Juanita is very interested in making sure that our observations are the observations are as accurate as possible. And even though the public will be involved, she's hoping that master naturalists will step up, and there'll be an opportunity for people to do maybe once a month observations up there at Cave Creek Regional Park.
Keep us informed of the volunteer opportunities and the timing of them and getting them on the calendar.
Right, we'll definitely do that once we got gotten everything in place.
Good.
Okay, if there's nothing further from the group.
I have one question. We're us cohort fours, our certificate said we're interns. So what more do we need to do?
Oh, yes, actually, thank you. I was actually wanted to say something about that. So I'm still extremely busy, thought my summer would not be as busy, but I've overcommitted yet again. So it would be really helpful if you think you're certified. So if you've reached your AT and volunteer requirement, if you think you've reached that, just shoot me an email and ask me to check. That would just help me tremendously. That way I don't have to sit there and go through everybody's records and look. Just shoot me an email and say, hey, I'm pretty sure I'm certified. I'll double check. And then what will happen is we will give you an additional certificate that says you're officially certified. And then we will also get your name tag ordered. Typically we were waiting till like the end of the year and doing like a bulk order, but they Lorianne and Deb said that it was fine to just order them as needed. So, and we'll just directly mail that to you. The company that prints them will just mail them right to you. And so you'll get that hard plastic name tag. So when you email me too, let me know what you would like on your name tag. You know, some people go by a shorter name or, you know, whatever. Just make sure we have your correct name and spelling that you would like on that name tag.
Okay, thank you.
Yes. All right, anything else? It was great seeing everybody. We had a lot of people attend. That's awesome.
Yes, very nice. And we know people travel during the summer too, but we're glad when you tune in.
Oh, and that was the other thing. Thank you, Kim. That jogged my memory. These are recorded, so we'll put these up on our YouTube channel. So once it's up, I'll try to remember to send an email letting everybody know it's there. But we do record each chapter meeting. So if you can attend, you can always watch it later, or if you want to go back and watch it again, it'll be there.
Another thing is, we will be starting our recruiting time for the next class. And by sharing the videos and letting other people, inviting people in, your friends and family to attend these meetings. We see this as a recruiting tool. So they are welcome to join us. The public is allowed to attend our meetings.
Great.
Oh, Amy's got a question.
Hey, so on that note, Kim, do you guys happen to have like a recruiting flyer that we could help distribute in our networks?
Oh, we've got a couple flyers and we've got some new ones because the cohort 4 made some to explain about the master naturalists, and Daniela did a
Daniela, yeah.
fantastic job with this booklet, spiral booklet, to explain what a master gardener, a master naturalist is,
Awesome.
and when we have table events too. So yes, we do have materials. And we,
Yeah. Sorry, I was
Go
just
ahead.
going to say that we are just about to open our Xeriscape demonstration garden at the City of Surprise City Hall. And we actually have like a bulletin board where we'll be able to put our water conservation workshops that are upcoming. And I would be happy to include like a recruiting ad on there for the Master Naturalist program, because I think we'll get a lot of people in the garden who are there because they're naturalists and some who are there because they're gardeners. So I want to be able to promote this program too.
okay, we will make sure that we get that to you.
I just put in the chat. Oops, nope, I sent it to Karen. Hang on. I put in the chat the brochure. If you want to print brochures, I don't know, Amy. I'm trying to say, I don't know how we can meet up, but or if you can meet up with Mo. Mo has a bunch that are already pre-printed. We also have the brochure in Spanish as well. I don't know if we have.
That's really cool.
Yeah, I don't know if we have a one-page like flyer. I mean, you're welcome to make something, like, you know, we don't have to like approve it or anything, you know, just make sure it has the logo
Okay.
on it. And you're welcome to type something up if you want, just like a, you know, something you can pin up. I was trying to find this.
I can do that. And if I create something, I'll put it on the Google Drive for other people too.
Yeah.
Amy, when was that garden opening?
It should be, as of right now, we're planning on sending out an official press release at the end of June. So basically right before 4th of July weekend, we'll be like, okay, it's open now.
So is this an event, or you're just gonna open it?
No, we're saving the event for the fall when it's cooler.
Smart
So we're
idea.
it's sort of like a soft opening via press release.
I have a question about Daniela's wonderful brochure. Is that available to be like printed and bound if I were to make, you know, is the like file available? Because up here in Flagstaff, there are a lot of people like me who live in the valley or elsewhere during the winter. And I'm sort of attached to the Colton Garden up here and the volunteers with the Museum of Northern Arizona and the Arboretum. And I feel as though it's a good recruiting base.
Good idea.
Yeah, so I was gonna say, actually, I just texted Danielle today about the booklets. Hang on one second.
I think I might try to, I know it's made in
Oh.
like no,
Are
I'm
you,
here.
are you
It's
on?
my-
I didn't think you were on. Okay.
Well, no, I've been on the whole time, but my video and like my screen is going in and out. I don't know why, but what I was gonna say is I know it's made like a certain like length and width, basically. I don't know if I could send it to you guys, or I
That'd
just
be
don't know what
great.
size you want me to send.
Well, and I was gonna say, so I'm actually, so I was asking Danielle to get me the files because we are gonna print some so that we have them for our, like tabling events and stuff.
Uh-huh.
So if somebody has a book that like they want to put somewhere, if you volunteer at an organization or whatever, and they want, and they'll allow us to have that booklet, if you let me know when I place the order, you know, we can get additional copies and have it paid for by the, you know, by our chapter. So, like I told Danielle, I had wanted just to like have a couple people proofread it, just make sure there's no like errors before we, you know, print a bunch of them. But yeah, if you, so if anybody has an idea of where we can put these, you know, let me know and I'll just add it onto the order. I priced some places today. Actually, rather than a spiral bound, I was looking at like a hardback book, kind of like a photo album type thing. And so I priced some of those today. So yeah, we can get those ordered for anybody that would like to put one somewhere.
Kathy, can we put it on a on the website, like as a booklet where people can flip through?
No.
Yeah.
It's almost,
Yeah, we can
almost
do
over.
that. Yeah. We're working, so we have a resource page. I don't know who just spoke, but we have a resource page.
It's Kim.
And,
We're done at 7:30.
we're going to
Be any minute.
be adding stuff to that, including links to capstone projects, because you know some people have done capstones that people can print, like somebody did like White Tank Mountain scavenger hunt and stuff. So we want to get these put onto the website, and now that we have somebody managing the website, yeah, I hope to get these resources up there so people have access to them.
Awesome.
All right. Well, go ahead, Jess, at 7:30. Oh, Andrea's, go ahead.
I just had sort of another question regarding having a brochure. I don't necessarily have a place to put a brochure, but I am willing to like either do my own table at the Colton Garden or, you know, take the a brochure and maybe Danielle's thing and show it to people where I know there are going to be you know, a bunch of potential contacts like Arboretum events that aren't like formal, but I know there are going to be a whole bunch of volunteers there. And the same thing at the Colton Garden and the M&A events.
Yeah, sounds good.
Okay.
Yeah, we're also going to order like business cards, just kind of generic business cards. So we'll have those, you know, and hopefully we can like have another get together and, you know, we can have some of the stuff available for people as well, like printed brochures. And so if you want to take like a stack of brochures and a stack of stickers and, you know, that way you have them with you. So we are planning on ordering some additional items now that we're growing, so people have things to hand out. So we'll have just like a generic business card that has like our website and, you know, our kind of main info email and things like that as well. So.
Terrific.
Are we officially doing our next in the fall rather than the spring like we did previously?
No, so we decided to stick with the spring, I believe. Is that right, Kim?
Yes, because we weren't ready to start again.
Right. Okay.
Right
Just
away.
I
So
knew.
it will be January.
Okay. Sounds good. Thank you. With that, no last minute questions. All right. Thank you guys all. I will not see you next month. Hopefully at some point.
Great.
Thank
you. Bye.
Good night.
Bye now.
Good night, everybody.
Bye. Bye.
Good night.
All right, I'll see you.
Okay. Glad everything went well.
Yeah.
Have a good evening.